There's a flower in Sumatra that spends 7 to 10 years hiding underground in a corm weighing up to 154 kilograms, 339 pounds. When it finally blooms, it erupts to over 10 feet tall in a matter of weeks. Its spadix heats itself to 37 degrees Celsius, 99 degrees Fahrenheit, the exact temperature of a human body. And then it releases putrescine, cadaverine, and dimethyl trisulfide into the humid night air. These are the same chemicals a decomposing human corpse produces. Carrion beetles and flesh flies arrive within hours, crawl inside, and unknowingly pollinate it. The bloom collapses within 24 to 48 hours. The plant may not bloom again for another decade. It will do this only a handful of times before it dies. In 1889, Kew Gardens grew the first one outside Sumatra. Thousands came. Some fainted. Today, the IUCN lists it as endangered. A flower that perfected the art of mimicking death is now actually dying. Indonesians call it bunga bangkai. Bunga means flower. Bangkai means corpse. The Western world calls it the corpse flower. Science calls it Amorphophallus titanum, which, translated from ancient Greek, literally means giant misshapen phallus. None of these names are exaggerations. This is the largest unbranched inflorescence on the planet. Not the largest flower. That title belongs to Rafflesia arnoldi, also from Indonesia, but the largest flowering structure that rises from a single stalk without branching. It is a biological skyscraper built by a bulb that sits underground in complete darkness, sometimes for a decade, doing nothing visible to the human eye. Storing energy, waiting. The plant was first scientifically described in 1878 by Italian botanist Odoardo Beccari. He was cutting through the equatorial rainforest of western Sumatra when he encountered something that defied his training. A single stalk taller than a man, wrapped in a deep burgundy spathe, radiating heat and producing a stench so overwhelming that it attracted swarms of insects from hundreds of meters away. He documented it. He sent samples to Europe. And for the next 11 years, every botanist who received those samples tried, and failed, to make it bloom outside its native jungle. Then, in 1889, the Royal Botanic Gardens at Kew, London, succeeded. The first corpse flower bloomed in captivity. Thousands of people lined up. Newspapers across England covered it. Some visitors reportedly fainted from the smell. The bloom lasted roughly 36 hours, and when it collapsed, the plant went dormant. It did not bloom again for years. To understand why this flower is so rare, you need to understand what is happening beneath the soil. The corpse flower does not grow from a seed into a flower and bloom in a single season like most plants. It grows from a corm, a massive bulb-like underground stem that functions as the plant's energy vault. The corm of Amorphophallus titanum is the largest known corm of any plant on Earth. The typical weight is around 50 kilograms, about 110 pounds. But the current world record held by the Royal Botanic Garden in Edinburgh is a corm weighing 153.9 kilograms. That is 339 pounds. It grew from a corm the size of an orange over 7 years. Each year, the corm sends up a single structure. In most years, that structure is not a flower. It is a leaf, but calling it a leaf is misleading. The leaf stalk, or petiole, can grow up to 6 meters tall, roughly 20 feet, and branch out to 5 meters across. It looks like a small tree. The petiole can be 15 to 16 inches thick at ground level. This leaf photosynthesizes for an entire growing season, feeding the corm, and then dies. A new leaf replaces it the following year. This cycle repeats for 5, 7, sometimes 10 or more years. Only when the corm has accumulated enough stored energy does it switch from producing a leaf to producing an inflorescence, the bloom. This is an all-or-nothing gamble. The plant commits its entire energy reserve to a single reproductive event that will last, at most, 48 hours. If pollination fails, those years of stored energy are wasted. When the corm finally triggers a bloom, the spadix, the tall central stalk, begins to rise. It can grow at a rate visible to the naked eye. The tallest documented inflorescence in cultivation was recorded at the Botanical Garden of Bonn, Germany, in May 2003, reaching 3.2 meters, over 10 feet 5 inches, measured from the corm. The Guinness World Record for the tallest bloom overall was set in March 2025 by Jane Whitechurch de Piero in Laguna Niguel, California, at 4.11 meters, 13 feet 5.8 inches. As the spadix reaches its full height, the spathe, the large petal-like sheath at the base, begins to unfurl. The outside is deep green. The inside is dark burgundy red, heavily furrowed, textured to resemble raw meat. Hidden inside the base of the spathe, invisible to the eye, are 2 rings of tiny flowers. The upper ring holds male flowers. The lower ring holds female flowers, spangled with bright red-orange carpels. The female flowers open first. This is critical. It prevents self-pollination. The male flowers will not release pollen until 1 to 2 days later, by which time the female flowers have already lost receptivity. The plant relies entirely on cross-pollination from a different individual. which in the wild may be kilometers away. To summon its pollinators across that distance, the spadix initiates thermogenesis. The tip heats itself to approximately 37 degrees Celsius, 99 degrees Fahrenheit, matching human body temperature. This is not metaphorical. The spadix becomes warm to the touch. This heat serves two purposes. It volatilizes the odor compounds, projecting them farther through the humid air, and it enhances the illusion that the spathe is a piece of warm, freshly dead flesh. The chemical cocktail that creates the stench has been analyzed by multiple research teams. It includes dimethyl trisulfide, which smells like Limburger cheese and rotting meat, dimethyl disulfide, garlic, trimethylamine, rotting fish, isovaleric acid, sweaty socks, benzyl alcohol, a faint sweet floral scent, phenol, like throat spray, indole, which smells like feces, and, as a Dartmouth-led study published in PNAS Nexus in November 2024 confirmed for the first time, putrescine, a biogenic amine produced when amino acids break down in decomposing animal tissue. The Dartmouth team, led by Professor G. Eric Schaller, used RNA sequence analysis and mass spectrometry on tissue samples collected from Morphe, Dartmouth's 21-year-old corpse flower, during multiple blooms. They found elevated levels of methionine, a sulfur-containing amino acid and precursor to the volatile sulfur compounds, in the spadix appendix during heating. Methionine levels dropped sharply within hours, confirming rapid conversion to airborne stench. The surprise was the detection of elevated putrescine precursors in the spathe tissue, the first molecular evidence that the corpse flower chemically replicates the specific decomposition signature of animal flesh. The odor peaks between late evening and the middle of the night, precisely when carrion beetles and flesh flies of the family Sarcophagidae are most active. The insects crawl into the base of the spathe, contact the female flowers, deposit pollen from another corpse flower they previously visited, and, deceived into believing they have found a carcass suitable for egg laying, stay long enough to pick up fresh pollen before leaving. By morning, the odor fades. Within 24 to 48 hours, the spathe wilts, the spadix softens, and the entire structure collapses. The show is over. The corm goes dormant. The next bloom may be 2 years away, or 10. According to Wikipedia, since the first cultivated bloom at Kew in 1889, there have been over 100 documented blooms in cultivation worldwide. But that number spans 137 years. In the wild, blooming events are even more rarely witnessed because the plant's habitat is deep, remote, equatorial rainforest. Some specimens in optimal greenhouse conditions, like those at the Botanical Gardens of Bonn, Germany, or the Copenhagen Botanical Garden, have been observed blooming every other year. But these are exceptions. Most cultivated corpse flowers take 5 to 10 years between blooms. The San Diego Botanic Garden notes that a corpse flower may bloom only every 4 to 6 years after the first bloom. Some remarkable cultivation events include the Bonn specimen that produced 3 simultaneous blooms from a single 117-kilogram corm in May 2006. The Chicago Botanic Garden achieved a triplet bloom in May 2020, nicknamed the Velvet Queen, although COVID-19 prevented public viewing. In 2011, Roseville High School in California became the first high school in the world to bring a corpse flower to bloom. The U.S. Botanic Garden in Washington, D.C. has displayed blooming corpse flowers in 2003, 2005, 2007, 2010, 2013, 2016, 3 blooms in 2017, 2 in 2020, 2021, and 2 more in 2022. Each event draws thousands of visitors and generates international news coverage. The plant's lifespan in cultivation is approximately 30 to 40 years. In that time, it may bloom as few as 3 to 5 times. Each bloom is a biological event that the plant may not survive if conditions are suboptimal. Amorphophallus titanum is listed as endangered on the IUCN Red List. Its native range is limited to the rainforests of western Sumatra, Indonesia, one of the most rapidly deforested regions on Earth. According to the IUCN assessment, the primary threats are timber harvesting and the creation of oil palm plantations, which have destroyed vast tracts of lowland rainforest where the corpse flower grows on limestone hills. There is also a local myth in some communities that the corpse flower possesses supernatural properties, leading to occasional tuber exploitation. The plant's own biology works against it. It requires years of energy storage before a single bloom. It relies on cross-pollination between individuals that may be widely separated. Its pollinators, carrion beetles and flesh flies, must be present in sufficient numbers during a window of less than 48 hours. If the forest is fragmented, if the distance between individuals increases, if pollinator populations decline, the probability of successful reproduction drops toward 0. A research paper from West Sumatra documented expeditions to find and characterize wild populations. The authors noted that the status of A. titanum is endangered because of several compounding factors: deforestation, tuber exploitation, extremely long intervals between flowering events, and protogeny. the fact that female flowers open before male flowers, making self-pollination nearly impossible under natural conditions. Botanical gardens worldwide have responded by building collections, sharing pollen, and developing advanced pollination techniques. This network of cultivated specimens may ultimately become the species' insurance policy. But a flower in a greenhouse in London or Washington is not the same as a flower in a Sumatran rainforest. Surrounded by the beetles and flies and microclimate, it co-evolved with over millions of years. The Dartmouth study, published in November 2024, represents the most complete molecular investigation of corpse flower thermogenesis and odor production to date. Professor Schaller's team collected 9 tissue samples over 3 nights from the lip and base of the spathe and the towering appendix of the spadix during Morphy's 2016 bloom. Exchange research scholar Alvina Zulfikar developed a method to extract high-quality RNA from the tissue, which is notoriously difficult in this species. RNA sequence analysis revealed elevated expression of genes encoding alternative oxidases, the plant equivalent of the uncoupling proteins that generate heat in animal cells. These genes were most active in the appendix tissue precisely when the spadix was heating up. Genes involved in sulfur transport and metabolism were simultaneously upregulated. Mass spectrometry on tissue extracts confirmed high levels of methionine in the appendix during the heating phase. Methionine is the metabolic precursor to the sulfur-based volatiles, dimethyl trisulfide and dimethyl disulfide, that form the core of the carrion scent. Methionine levels plummeted in samples taken just hours later, confirming rapid enzymatic conversion and volatilization. The unexpected finding was elevated levels of a putrescine precursor amino acid in the spathe tissue. Putrescine is a diamine produced during the decomposition of animal protein. Its presence in the corpse flower's scent profile was known, but this was the first study to trace its genetic and metabolic origin in the plant. The spathe, the structure that visually mimics meat, is also the structure that chemically mimics decay. Form and function are perfectly aligned. Professor Schaller noted that the plant essentially runs the same metabolic pathways that a decomposing animal body runs, but in reverse. Instead of putrescine being a byproduct of death, the plant synthesizes it deliberately, on demand, at precisely the moment it needs to attract pollinators. If you want to see a corpse flower bloom in person, here is what you need to know. Botanical gardens that maintain active corpse flower collections include the United States Botanic Garden in Washington, D.C., the Huntington Library and Botanical Gardens in San Marino, California, the New York Botanical Garden, the Chicago Botanic Garden, the San Diego Botanic Garden, the Royal Botanic Gardens at Kew, London, the Botanical Garden of Bonn, Germany, the Copenhagen Botanical Garden, and Dartmouth College's Life Sciences Greenhouse in New Hampshire. Most of these institutions maintain bloom watch pages or email alert lists. When a corpse flower begins to show signs of blooming, the bud tip emerges from the soil and grows rapidly. Gardens typically announce it days or weeks in advance. Once the spathe begins to unfurl, you have 24 to 48 hours. Some gardens stay open through the night during bloom events. Expect crowds, expect a line, expect a smell that most visitors describe as rotting meat, old fish, dirty socks, and death all at once. And expect it to be worth it, because the next bloom at that institution may be years away. For those who cannot visit in person, many gardens live stream bloom events. The U.S. Botanic Garden and the Huntington have both hosted widely viewed webcam streams during recent blooms. This is a flower that takes 7 to 10 years to build a single bloom. It heats itself to human body temperature. It manufactures the chemical signature of a rotting corpse, putrescine, cadaverine, dimethyl trisulfide, not by accident, but through deliberate genetic pathways that were only decoded in 2024. It fools beetles and flies into pollinating it during a window of less than 48 hours. Its corm can weigh more than 300 pounds. Its spadix can tower over 13 feet. It will do this perhaps 3 to 5 times in 30 to 40 years of life, and it is going extinct because the Sumatran rainforest it evolved in is being cut down for palm oil. A flower that spent millions of years mastering the art of smelling like death is now, for the first time, actually facing it.